So I'm just going to talk a bit about e-field modeling and current flow models and whether they're fit for purpose. And the purpose, of course, is that they help us to optimize the outcomes of stimulation, to make things better, get better effect sizes, and so on and so forth. And the basic idea is very simple. Based on what we know about anatomy and conductivity and so on and so forth, can we predict what we're actually delivering to the brain? So that's a very sort of simple idea, I guess. Next slide, please. And the reason why we want to do that is that I would say or argue that pretty much for all types of non-invasive brain stimulation, we really bedeviled with a lot of variability in the effects and often poor reproducibility and small effect sizes. So that's a real problem, especially if we want to go to translation. Next slide, please. And so perhaps one big factor contributing to this variability is that generally for a lot of stimulation protocols, we apply fixed stimulator output to all our subjects, even though we know that that will lead to highly variable delivery of stimulation to a specific brain area or network. And so that is likely to at least be a big contributor to variable effects. So if we could use modeling, we might be in the position to adjust the stimulator or the delivery so that everybody receives the same stimulation in a target. And that's, of course, a very appealing motivation. Next slide, please. So generally, I think, or in the majority of cases, we would probably use these models to control the intensity of current, the electrical field in a target area, for example. And that has been done in a lot of cases. But we also know that the spatial layout of the current we deliver is important in terms of determining the physiological and behavioral effects and the direction of current with regard to the anatomical structure or neurostructure that we're trying to target. And this is probably, these 2 points are much less often considered. Next point, next slide, please. And so if we turn to intensity, it's been now shown, I think, in quite a lot of studies that in cases that we deliver the same stimulator output to our population, the actual electrical field delivered to a target area, say motor cortex, can vary by 100% or more across our subjects. And so clearly that is an issue we need to address. And with modeling, we can clamp this and we can essentially eliminate this variability, at least at the target site. Now, the reason why that is an issue is that even if you use exactly the same stimulation protocol in everyone, just because we're sampling from a population and the anatomy really determines what we're going to deliver, we might end up with 2 studies that deliver something quite different, even though they try to fix everything. And the smaller the sample size, the more severe this problem becomes. Next slide, please. But so even if we fix this parameter and we try to deliver the same electrical field intensity to the target in everybody, we will probably change the spatial configuration or the focality of stimulation and the amount of cortex or brain tissue that it is exposed to a particular electrical field above a certain threshold. Now, we could turn, in the case of TES, we could turn to something like HD TDCS, but even there, we have the problem that this is not a focal stimulation technique. And so we're not quite sure what, or we can't easily control what is actually delivered to all the other brain areas outside our target area. Next slide, please. And then on top of that, we've known for a long time that the direction of current with regard to our neural structures is of great significance to what it actually does to those neurons. Now, in the case of TES, we have the problem that we have a folded cortex, and so at any point that is exposed to current, we may actually get very different polarization of the neural structures we want to target. And this is true for very nearby areas, such as adjacent areas in the sulcus or the gyrus versus the bank of the crown versus the bank of a structure. So again, this is not that often considered, and we don't quite know how those effects will lead to the consequences we observe. Next slide, please. So here we show this in just two examples where we consider the direction. So what you see here color coded is basically the direction of current flow with regard to the cortical surface. And what you see is that in very nearby structures, so here, for example, in primary and sensory sensory cortex, we may get actually opposite direction of current. So radial inward with radial outward currents. And we get perhaps very different current direction when we consider the crown versus the bank of a sulcus or gyrus. So in how this all conspires to give us the physiological and behavioral effects that we observe, we don't know. And next slide, please. And so you can look at that across a population for different montages and so on and so forth. And basically what you see is that the direction of current with regards to the cortical surface is highly variable depending what we do. But we don't know what that actually does to the brain. Next slide, please. So I would argue that E-field modeling and current flow modeling is a necessary requirement for us to turn to stimulation and make it more effective. But in a lot of cases, we actually don't know what we should control for. Is it just the intensity of the electrical field, its direction, its focality, the spatial layout, the amount of cortex exposed to a certain electrical field? And it's not an easy problem to solve, because as you tweak some of these parameters, the others change as well. And we can perhaps do this, but then these models become increasingly complex. And I think then the problem is that their use becomes very limited, certainly in sort of some research settings and also in widespread clinical applications. And so we need to ask if the increasing model sophistication is really giving us better predictions. So ever finer sort of anatomical models and so on and so forth, do they actually help us getting better outcomes compared to what we had 10 years ago? And I think what was nicely illustrated in the previous talk is that we're not really interested in E-fields per se. What we really want to know is what do they do to the brain. So what we really need is we need to link these models, again, as was nicely argued in the previous talk, to models that tell us something about the physiological impact of what current does, and that tell us whether a target is indeed engaged and what the physiological consequences are. And at this stage, I think all of this is still quite cutting edge and certainly not mainstream and not widely used. Next slide, please. So the real challenge is therefore to go from the stimulation to predict where and we deliver current to and how much, to predict what the physiological consequences are. But really what we want to know is how this physiological change is changing the computations in a brain area and therefore the behavior, because that's what we all want to actually modify with stimulation. Next slide, please. And so one really big challenge of this is that any type of stimulation is likely to alter quite a lot of different, if you like, mechanisms at different levels of observations, and we often we're not able to predict what that will be. And then the big problem is that in many cases, these consequences will be different in different pathologies, and we often can't predict what that consequence will be. Next slide, please. Now that will be fine if, say, the change in pathology and how it affects the state of the brain is simply leading to a linear change, like here shown in panel A. But probably in a lot of cases, the pathology or the abnormal state, say, can have nonlinear consequences on what stimulation actually achieves. And if we can't predict that, we essentially end up in the situation where we can't predict at all what's going to happen, and maybe sort of some unforeseen consequences occur. And again, I think the previous talk made it really clear that in cases like epilepsy and so on and so forth, that's really a fundamental problem to solve. Next slide, please. Now, if you turn to something like stroke, which is shown here, then that's even another clear example. So here we don't just have state changes in and around the network we're targeting, but we also have structural changes. And as shown here, they can profoundly influence, in the case of TDCS anyway, how current is delivered to the brain and where to. Depending on the size and location of the lesion, we might have variations of up to 30% upwards or downwards to what we would have in a healthy brain. And next slide, please. So are current flow models fit for purpose? I think the answer is maybe. They already do, or e-field modeling does a great job in predicting e-fields, and I'm not sure how much more we have to squeeze out of these models in terms of do they give us actually something that much better. But we often don't know what to optimize for. We think that in a lot of cases we might really need to turn to these specific models. That's certainly dependent on the pathology. But critically, we need to couple those models with physiological models, and ultimately with models that help us to predict how the physiological change changes the behavior we're interested in, whether that's in a patient or not. And with all that and the exciting developments, as we just heard, ideally those models should remain usable. So that's perhaps that requires squaring a circle, but it's certainly something we need to bear in mind for their usability. Thank you very much.